All right, let's just start right now. I would like to extend my welcome to everyone who has joined till now. And please just let me know if you can hear it and whether the resolution is okay and whether you can hear my voice. And let's then begin without any further ado. Is the mic being set up right? Am I audible? All right. Okay. So let's just start. Today we'll be reviewing renal physiology, and this might be a part of a two-part series, whether we'll be going over the basics of renal physiology, and we shall also be studying some of the clinical correlates related to renal physiology. And at the end of the discussion, I just believe that we can make this stuff a lot easier than you might expect. And just like you guys, I'm also preparing for my NEET PG examinations, which are due next year. So let's just get something out of this session right now, right? And I'm very free to questions. If any question arises, whether you want to uh, have me repeat something, you can very well do that. All right, so let's just start. So first of all, we'll be looking over a basic structure of a glomerulus. As we all know, that a glomerulus is a tuft of capillaries and it arises from the afferent arteriole and then it ends even into an efferent arteriole. And this is the basic layout of a glomerulus. As you can see, first we discuss about something known as epithelial cells. We have two types of epithelial cells. We have the parietal epithelial cells and we have the visceral epithelial cells. The parietal epithelial cells are just about right here, and these are the visceral epithelial cells which extends these foot processes known as podocytes into the glomerular capillary, and they make a junction over there, right? We have the mesangial cells. We'll be discussing about the mesangial cells in great detail, how we can have different types of mesangial cells. We can have glomerular type of mesangial cells, or we can have extraglomeralar mesangial cells, which are rightfully known as Lassus cells, right? So these are the podocytes. They're extending their foot processes, and this is the fenestrated endothelium. What I mean by a fenestrated endothelium is that these endothelium, they have fenestrations within them. So there is a passage of solutes which can occur through these endothelium as well. And this right here is the basement membrane, and this structure, that whole structure, is known as a glomerulus. So we have the endothelium, we have the fenestrated endothelium, we have the basement membrane, we have the foot processes that these podocytes extend, we have the visceral parietal epithelial cell layer, and between the parietal and the visceral epithelial cell layer, we have the Bowman space, also known as the urinary space, that is between the parietal and the visceral epithelial layer. Now we shall be looking over in great detail, and let's just magnify this portion right now. And here we go. So this is the foot processes of the podocyte that we saw recently, and this is the basement membrane. And this basement membrane has actually three subsections. We have the lamina rara externa, we have the lamina densa, and we have the lamina rara interna, right? So the lamina rara interna faces towards the endothelium side, and the lamina rara externa faces towards the podocytes. So here we have the podocytes, we have the basement membrane, we have the fenestrations in the endothelium. These fenestrations are 70 to 90 nanometers big. As you might see, that seems to be a problem because we can't allow particles which are of much higher size. We'll be discussing very shortly that we can have particles up to the range of 8 nanometers. Above 8 nanometers, none of the solutes can pass through it. And within 4 to 7 nanometers, we have we give preferences to those solutes which are positively charged, followed by neutral solutes, and followed by negatively charged proteins. We'll be discussing over that shortly. So that is the thing. We have the fenestration, they 70 to 90 nanometers big. We have the podocytes, and here we have a structure known as the filtration slit. As you can see, there are different proteins which are involved in the filtration slit. We might have the protein known as nephrin, then we have podosin, then we have CD2AP, that is a CD2 associated protein. We have some actin oligomers which extend from the CD2 associated protein, and we have NEF. Why are these proteins so important, you might ask? Because many of these proteins undergo mutations. For example, a characteristic example would be nephrin, right? So nephrin undergoes a mutation in something known as the congenital nephrotic syndrome of Finnish type. So that is clinically very important for us that we remember this structure known as the filtration slit. Now, the diameter of the filtration slit is 4 to 11 nanometers, which is significantly shorter or significantly narrower than the fenestration. They were 70 to 90 nanometers long. And a very important component of this basement membrane is collagen type 4. And here you can see collagen type 4. And why is this also very important? Because we should know that the collagen has a triple helical structure. It has some domains. We have the globular non-collagenous NC1 domain, we have the 7S domain, and we have the triple helix protein. Why is this so important? We'll be going over very shortly. But just remember, collagen type 4 is present in the glomular basement membrane. Let's move in front. So this is the particular structure of a nephron. As we saw, we have the afferent arteriole, we have the efferent arteriole exiting, we have the glomerular capsule over here, we have the proximal convoluted tubule over here, and right now we'll be discussing over this. This is the loop of Henle, and this is the thin descending limb, and this is a thick ascending limb. Sometimes we even have a thin ascending limb, and we'll be discussing the differences between a cortical nephron and the juxtamedullary nephron, where things will be much clearer. Then we have the distal convoluted tubule, which empties itself into the collecting duct. So we have different DCTs entering into the collecting duct, and then ultimately the collecting duct opens into the renal pyramids, right, and ultimately into the calyces. So this is the basic structure of a nephron, right? So we have very importantly, we have very two stark type of nephrons with different functions over here. So we have cortical nephrons and we have juxtamedullary nephrons. So let's just go over the differences between them and let's just see them in a diagrammatic manner later on. So we have cortical nephrons. Cortical nephrons comprises about 80%. Some books have 85% written. So it's 80% of every nephron present in a body. And juxtamedullary nephron contribute about 15 to 20%, right? So these cortical nephrons are present in the renal cortex. And where in the renal cortex? Much closer to the outer side of the renal cortex, right? Cortical nephrons have very short loop of Henle. They do not extend deep into the medulla, right? And so all of these nephrotic segments of the cortical nephron are restricted to the cortex, right? And one more difference between a cortical nephron and a juxtamedullary nephron is there is no ascending thin limb, which we discussed over here, that we might have a thin ascending limb over here. So this might be a cortical nephron, right? On the other side, we have juxtamedullary nephron, which comprise about 15 to 20% of all nephrons in a body, right? The glomeruli of these juxtamedullary nephron, in contrast to the cortical nephrons, they lie more towards the cortex, which is closer to the medulla. So they lie in the deep cortex. They have long loops of Henle as compared to the cortical nephron, which have shorter loops of Henle. The juxtamedullary nephrons, the loop of Henle of the juxtamedullary nephron goes deep into the medulla, while most of the nephronic segments of the cortical nephron were restricted to the cortex itself, right? Cortical nephrons have a very extensive network of peritubular capillaries present within them. We'll be seeing them shortly. And the juxtamedullary nephrons, on the other hand, have very specialized peritubular capillaries, which are known as vasa recta, right? So one of the main differences between cortical nephron and juxtamedullary nephron associated with their function is the cortical nephrons, they basically contribute to urine formation, whereas juxtamedullary nephrons, they basically concentrate the urine, right? So basically, urine concentration involves the removal of water from the urine or from the tubular lumen and adding them into the venous system later to be taken on further. So let's just see diagrammatically what do I mean by that. So let's just see, this is a cortical nephron, right? So how can I say that it's a cortical nephron? First of all, just mind this, there is no thin ascending limb, a very important thing. As you can see, we have a sheath of capillaries surrounding each of the nephronic segment, and these are known as peritubular capillaries, right? We have the loop present over here, and all of these nephronic segments will be restricted to the cortex itself. Or I can say that the glomerulus of the cortical nephrons lies towards the outer cortex. And what is the function of these cortical nephrons? It is urine formation. Let's move on to the next set of nephrons. That is the next one that we have discussed. Next we talk about the juxtamedullary nephrons. So the juxtamedullary nephrons, like we can see the word, they're juxtamedullary, close to the medulla, right? So they lie towards the inner cortex of the kidney. And they have a specialized peritubular capillaries, which are known as the vasa recta, which facilitate countercurrent exchange. We'll be going over that countercurrent exchange mechanism shortly. So basically, they facilitate the vasa recta as basically peritubular capillaries, which are arranged in a parallel manner, and functions as a countercurrent exchange mechanism. And as you can see, these guys have a thin ascending limb. They're going to have a thin ascending limb. So that is the difference. We discussed about the structure of a nephron. We discussed about the structure of the glomerulus. Now let's move front. Right. So before discussing further, we should remember what is the juxtaglomerular apparatus. So the JGA, or the juxtaglomerular apparatus, consists of three main components. So one of the components are the granular cells. They are specialized smooth muscle cells present in the afferent arteriole, which basically do not contract, but they basically secrete a protein known as renin. And renin, as we all know, is very important in the renin-angiotensin-aldosterone system. So this participates in the RAS mechanism, right? The next in line, so these are the JG cells. They're specialized or modified smooth muscle cells, also known as granular cells, and they release renin. The second one that I would like to draw your attention to is the macula densa. As we all know, all of the tubules, they have an epithelium, and these epithelium are cuboidal, right? But the macula densa is columnar epithelium. So this is the portion of the nephron which has columnar epithelium as opposed to the cuboidal epithelium present everywhere else. So this macula densa, where is it located? It is located just at the junction of the thick ascending limb and the distal convoluted tubule. So this is the macula densa, right? And the third group of cells are the Lassus cells, which we discussed, right? We have two types of mesangial cells. We have Lassus cells and we have the interglomerular cells. So Lassus cells are nothing but they're extraglomerular cells, right? So this is the juxtaglomerular apparatus. One more, recently examiners would like to ask you this question. Like, there is something known as the calcium paradox. Usually when we talk about calcium, we talk in terms whether it can facilitate contraction, right? Or it can lead to exocytosis, right? So calcium mediates exocytosis, as you might have all studied, under vesicular transport, that it binds to proteins known as snares, right? If I have to be very specific, it is synaptotacmin that it binds to. So as we all know, calcium means if there is an increase in calcium via the phospholipase C pathway, it leads to exocytosis wherever in the body, except here. So the renin cells, they have something known as the calcium paradox, in which we see whenever there is an increase in intracellular calcium, there is a decreased secretion of renin. Guyton clearly says that there is no particular mechanism why this happens, so we have to go with that. So this is an unknown mechanism, calcium paradox, seen in the JG cells, also known as the granular cells, right? As we all know, so there are particular events in a nephron. So we can have filtration, like whatever blood is flowing through the glomerulus, it filters into the Bowman space, which we saw that was the structure for the space between the visceral epithelium and the parietal epithelial layers, right? And then we can have some reabsorption by the tubular lumen by the different transporters which we'll study, and we can have some secretion into the tubular lumen, right? So this is the basic stuff. Now let's move on to the functions of the kidneys in front. So first thing is excretion. So it leads to excretion of these nitrogenous wastes. Hello, am I audible? Please, I would like to have an update, please. Is it audible? I'm waiting for someone to type. All right. Okay. So what are the different functions of the kidneys? So one thing is the excretion of nitrogenous waste like urea, uric acid, and creatinine, and other toxins are present in our body. And second is the excretion of bioactive substances like xenobiotics or drugs, right? The second function of the kidney is regulation. What does it regulate? The regulation of blood pressure through renin. We saw the renin-angiotensin-aldosterone system, long-term mechanism of your regulation of blood pressure. Second, we have the regulation of the acid-base balance, which we'll discuss in another video that's about acid-base balance. And third is regulation of the water and the electrolyte balance present in our body. The third major function of the kidney is production. What does it produce? It produces erythropoietin. And very favorite MCQ, where is the erythropoietin secreted from? It is secreted from the interstitial cells of the peritubular capillaries, which we just saw. So we have production of erythropoietin, which works on the RBC precursors, and that leads to increased synthesis of RBCs. Second, production of glucose, gluconeogenesis. Don't forget, glucose 6-phosphatase enzyme is present in the liver and in the kidney. So it functions in gluconeogenesis, right?
And the third major function is production of vitamin D by production of 125 dihydrocholecalciferol, right? So this is we know the nephron is the functional unit of the kidney, and each human kidney has approximately 1 million nephrons, right? And we know that each nephron contains a tubule and a glomerulus, and we saw the different portions of the nephronic segments, as well as we discussed the differences between your cortical nephrons and juxtamedullary nephrons. So again, the differences again summarized. So we can see the renal corpuscle is made up of the glomerulus. We have the glomerular space which surrounds the glomerulus, and it has a parietal layer, it has a visceral layer, we talked about it. The total length of the nephron is somewhere about 45 to 65 millimeters. So now we talk about the different parts of the renal tubule. We have the proximal convoluted tubule, we have the loop of Hille, we have the DCT, and we have the collecting tubules, right? And we will discuss the different portions very frequently. So this is again a structure. It contains, we have the cortex, and we have the medulla, and we have the renal pyramids. So these are the pyramids, and each of these collecting ducts empty into the your minor calyces, and later into the major calyces, right? And ultimately into the renal sinus, right? So this is again the structure. As a very important thing is the arterial supply. We should not forget the arterial supply of the kidney. So just whenever the kidney, so here is the structure of the kidney, and the arterial supply of the kidney arises from the aorta at the L1, L2 level. And whenever the renal artery arises, it gives two divisions. We have the anterior division and we have the posterior division. And later on, these forms the segmental artery, which basically supply the different segments of the kidney. So what are the different segments of the kidney? We have the apical, upper, middle, lower, and a posterior segment of the kidney, right? And at the junction of these lines, we have the Broden's line, which is very important surgically because there is an avascular plane during which we perform nephrectomy. So this is the Brodel's line between the anterior division and the posterior division of the renal arteries, right? After giving these segmental arteries, we move further. So after giving the segmental artery, we have something known as the lobar arteries. The lobar arteries then later on give the interlobar arteries. These interlobar arteries basically then form the arcuate arteries. So these are the arcuate arteries. These arcuate arteries later give on interlobular arteries. So we move down to lobar. Lobar, say we move down to the interlobar, then the arcuate. Now we're on to the interlobular. And the interlobular artery gives into the afferent arteriole, and later on we know what happens, right? So this is the site. This is the site of the Brodel's line. So this is the Brodel's line. This is the avascular plane. So this is the anterior portion, and this is the posterior portion of the kidney. And along this line, do we go for nephrectomy, right? Junction of the medial 2/3 and the lateral 1/3 surgically for performing any nephrolithotomy, for removing any stones, right? Right, so we discussed about this. We discussed about the filtration slit. We have discussed about the fenestration, 70 to 90 nanometers in diameter. And we discussed about the basement membrane and its major subsections, right? And we have the foot process of the podocytes, which we have already discussed. And we also discussed something about the filtration slit and how does it contribute in narrowing down in the fenestrations, right? And different proteins which are associated with the filtration slit, that is neph, nephrin, CD2AP, that is CD2 associated protein, as well as podocyte. So the JGA, we've discussed the major functions. We've discussed about the JG cells, also known as the granular cells, and they secrete renin. The macula densa, will discuss how it senses sodium in the tubular lumen, and this is a fantastic mechanism which you'll be knowing, right? That is the tubuloglomerular feedback. We'll discuss about that as well. And then we have the extragranular mesangial cells, which you know are known as the Lassus cells, right? Right. So we discussed about the major function. We have the glomerular filtration. This is filtration. And then we have the tubular reabsorption. We sometimes even have tubular secretion, and then ultimately urinary excretion in the form of urine. So let's talk about the glomerular filtrate now. So as we discussed, up to 4 nanometers in size, the 4 nanometers is very important. 4 nanometers, up to 4 nanometers, any particle, yeah, or any solute of any charge can pass through. That is only up to 4 nanometers. 4 to 7 nanometers, it depends upon the charge. 4 to 7 nanometers, it depends upon the charge. How? Because the basement membrane, it contains some proteins. Also, it contains the glycocalyx, which is negatively charged. It has some hyaluronic acid, which is negatively charged. So we know the basement membrane, it basically is negatively charged, so it will repel negative charges. So anything that is 4 to 7 nanometers,
negatively charged. If the solute is negatively charged, it will be repelled and it will pass with less ease as compared to those which are neutral or positively charged. So there's a differentiation between the size 4 to 7 nanometers. With negatively charged proteins like albumin, for example, albumin is 6 nanometers in diameter. So it lies between 4 to 7 nanometers. Because albumin is negatively charged, it is repelled. And that is why we have some amount of protein being lost in the urine, but not excessively, like 150 mg per day in 24 hours. That is perfectly normal. Then 500 to 400 is functional proteinuria. So basically, albumin is basically repelled due to its size as well as due to its charge. So that's the main thing. Anything more than 8 nanometers, it can't be filtered out. So the glomerular filtration can't filter large proteins due to their size and the repulsion between the proteins' negativity, such as albumin and negatively charged glycoproteins like type 4 collagen and proteoglycans, right? So let's just talk about the glomerular filtration rate. So the different portions which contribute to glomerular filtration rate, we'll be discussing them shortly. You have something known as the glomerular hydrostatic pressure, which is nothing but the pressure with which the glomerular capillary is being perfused. So we represent it as PC. So this is the glomerular capillary pressure. So this amounts to be 60 millimeters of mercury. Then we have the plasma colloid oncotic pressure, also known as the plasma osmotic pressure. That is the oncotic pressure. That is due to the proteins which are present in the glomerular capillaries. So this plasma colloid oncotic pressure amounts to 32 millimeters of mercury. So this is a blood vessel. For example, if I'm drawing this, so the forces which facilitate the glomerular filtration include the PC, that is the hydrostatic pressure present in the capillaries. The thing that is repelling it is the pi c, that is the oncotic pressure between the capillaries, right? So this is about the capillary, whatever forces, whatever the stalling forces which are acting, which either facilitate filtration or they repel it. The second thing that facilitates this glomerular filtration is your pi if, that is the oncotic pressure present in the interstitial fluid, that is in the Bowman's capsule. And the thing that is repelling the filtration is your PIF, that is the oncotic, hydrostatic pressure present in the interstitial fluid. So these are the different stalling forces. So whenever we amount to them, for example, the PC is about 60, the pi if is 0, the PIF amounts to about 18, and the pi C amounts to be about 32 millimeters of mercury. So if you combine these things which are opposing filtration, they amount to be 50. And the thing that is facilitating filtration, that is your glomerular capillary hydrostatic pressure, that is about 60. So the net filtration pressure becomes 10 millimeters of mercury, right? So this is net filtration pressure, that is 10 millimeters of mercury, right? So how do we calculate the GFR then? So we have the net filtration pressure, we multiply it with something known as the capillary filtration coefficient, right? That is the KF, capillary filtration coefficient. So we multiply the KF times the net filtration pressure. So let's do that. So we have 12.5 ml per minute per millimeter of mercury. That is the capillary filtration coefficient. We multiply it by 10. And you guessed it right, that it is 125 ml per minute. So GFR 125 ml per minute, which will amount to be about 180 liters of glomerular filtration occurring every day, which is much more than the plasma volume of only nearly 3 to 4 liters, right? So this is the filtration coefficient. We have something known as the filtration fraction. So what is this filtration fraction? So we have the renal blood flow, which will be later be seeing how do we measure renal blood flow, how we measure GFR. So the renal blood flow, which we measure by para-aminocupric acid, that amounts to be about 25% of the cardiac output. The cardiac output is 5 liters per minute. So 25% of the cardiac output, that amounts to be about 1250 ml per minute. So the renal plasma flow, which is calculated from the renal blood flow, as we all know by the formula, that the renal blood flow is RPF divided by 1 minus hematocrit, right? So the renal plasma flow comes to be about 620 to 650 ml per minute. So the filtration fraction becomes how much of that blood which is being sent into the kidneys in the form of plasma is getting filtered out as GFR. So we divide it and we get a fraction. This fraction is known as the filtration fraction. So GFR divided by the renal plasma flow, that is 125 ml per minute, divided by 625 ml per minute. So this amounts to be about 1/5, that is 20%. So the filtration fraction amounts to be about 20%. Next, we move on to what is the GFR is governed by. So we have the total surface area of the exchange that is happening. So that is one thing. The second thing is the filtration of the membrane permeability, which is all dependent on the capillary filtration coefficient. So these two things, that is the total surface area as well as the membrane permeability of the nephron, is dependent on the capillary filtration coefficient, which is calculated to be about Kf was 12.5 ml per minute per millimeter of mercury, which we multiplied by the net filtration pressure of 10, which were the pressure exerted by the Starling forces, right? So the GFR amounts to be about net filtration pressure into the Kf, right? So the Starling forces are the forces that control movement of fluid in or out of a capillary. And we studied the different Starling forces. We can have hydrostatic forces, so the hydrostatic pressure, that is which pushes the fluid outside, or we have the colloid osmotic pressure or the oncotic pressure, which pulls the fluid inside, which is mainly due to the proteins which are present in the glomerular capillary. So this is again a diagrammatic representation what we were talking about. The net filtration pressure amounts to about glomerular hydrostatic pressure, which is 60, minus the sum, or we can take it as a sum, Bowman's capsule pressure, that is the PIF, and we add it to glomerular oncotic pressure, which is your pi C, and this amounts to be about 50. This is 60, so the net filtration pressure comes to be about 10 millimeters of mercury. So now, as we have studied GFR, now we'll be talking about the regulation of GFR and the renal blood flow. Before we discuss that, we should be very familiar with the thing known as autoregulation. This happens in the heart, and this happens in many different organs, and one of those organs is your kidney. So what is the meaning by autoregulation? So the autoregulation means one organ is able to control its own dynamics. That is known as autoregulation without any external forces, right? Let me give you an example. For example, in the kidney, we have some mechanism through which we can alter the GFR and the renal blood flow in the kidney. For example, even let's take it one more further. We have the heart rate, right? We can have some forces which can influence the heart rate from outside, right? So that do not contribute to your autoregulation. That is external forces which are acting on the heart. Then we can have some structures which are present within the heart which autoregulate it. So that will be termed as autoregulation, right? So what is this autoregulation all about? So the renal blood flow and the GFR remains constant over an arterial pressure which is present anywhere between 80 to 170 millimeters of mercury. Some books state to be about 90 to 220 millimeters of mercury. I think this is a better range, 90 से लेके 220 millimeters of mercury तक the renal blood flow or GFR will not change, even if the arterial pressure changes. Now, how does the kidney manages that, which we'll be studying? So you have two mechanisms. There's a tubulo-glomerular feedback, which we'll talk about, and we have the myogenic autoregulation of renal blood flow. So one is very easy. So let's discuss the easy one first. That is a myogenic mechanism. So what is the myogenic mechanism? The myogenic mechanism is very easy. For example, whenever you have increase in pressure, so there is this is a blood vessel, and it is experiencing some change in its pressure. So this is the PC. We have an increase in pressure in the capillary. So what happens is we have these smooth muscle cells present in the afferent arteriole. So whenever there is an increase in pressure, so these smooth muscle cells, they have stretch sensitive calcium channels, right? So these are the stretch sensitive calcium channels. So whenever there's an increase in pressure, this stretch sensitive calcium channels will sense that pressure and they will contract. And once they will contract, they will decrease the GFR, right? So that is the main thing. So the myogenic mechanism is easy. So the next mechanism that we're talking about is the tubulo-glomerular feedback, right? And we know the tubulo-glomerular feedback was basically in turn due to the juxtaglomerular apparatus, right? So let's just see what do we mean by tubulo-glomerular feedback. Let's just assume there's a decrease in a renal arteriolar pressure. So the glomerular hydrostatic pressure also falls, and we are experiencing there is some decrease in the GFR. Now the renal autoregulation has not kicked in right now. We'll be discussing how does it kicks in. So what happens is, whenever there's a decrease in GFR, there's a decreased solute reabsorption in the proximal tubule. There's a decreased solute reabsorption in the thick ascending limb. And ultimately, because the GFR is less, there is less sodium in the tubular lumen. So there is less sodium in the tubular lumen that reaches less towards the macular densa. So you can imagine the tubular lumen has less of sodium, and here is the macular densa doing its job, right? How does it function as a chemoreceptor? Let's just talk about that. So there's a decrease in the NaCl. If there is a decrease in the sodium chloride levels in the tubular lumen in the macular densa, what happens is there is decreased sodium reabsorption through the macular densa. And we all know that sodium reabsorption in many parts of our body, especially the kidney and the GIT, works by secondary active transport, right? Matlab, whenever there is a reabsorption of sodium by the macular densa cell, there is often the use of 3 sodium and 2 potassium, that is the sodium potassium ATPase, generating the concentration gradient against which the sodium can move inside the tubular lumen, right? So whenever there is a decrease in the macular densa NaCl, there is decreased need of the sodium potassium ATPase to function. So whenever there's a decreased need of the sodium potassium ATPase to function, what will happen? There's decreased consumption of ATP. So there is decreased ATP consumption. So this decreased ATP consumption would mean that the basic substrates which break down ATP breaks down to, that is your adenosine and ADP, they will decrease. So adenosine and ADP levels will decrease. So once these adenosine and ADP levels decrease, what happens is, let's just see this diagram. So you have a decrease in the adenosine ADP and the adenosine levels. So you have the JG cells over here. So the JG cells have the receptors against adenosine and ADP. So we have the P2Y receptor for ADP and we have the A1 receptor for adenosine. Whenever these receptors are stimulated, they lead to increase in calcium. But what are we talking about right now? We are talking about these substrates are less. So there is decrease of ADP and there is a decrease of adenosine. So whenever there is a decrease of ADP and there is a decrease of adenosine, that will not cause these receptors to get stimulated and there will be a decrease in calcium. And what was the calcium paradox, my friends? The calcium paradox was that the JG cells, they basically release more renin whenever the calcium is low. So now whenever the GFR was decreasing, we started with the GFR being decreasing. Now what do we have done? We have increased the renin. We have increased renin angiotensin system. We have activated the RAS mechanism, right? So this is the beauty of the tubular-glomerular feedback. How does it manages to do that? Okay. So you can go through the slides and we'll be short of time and we'll discuss it further. Okay. All right. All right. So let's just discuss the different determinants of the GFR. So, all right. So I think guys, today it would be enough. So we'll be continuing rest of the section. We've covered almost about 28 slides. We'll be going over the next of the slides later, right? In tomorrow's discussion, right? All right. Thank you everyone for joining. And how was the session? Please, anyone tell me about this. Is my first time doing this kind of live session, and I understand the connection might be slow, or there might be some problem in viewing the slides. I can improve that by tomorrow onwards. And how did you guys enjoy the live?